Hey everyone, this is video number 3 for module 6, and this module is about gene linkage, recombination, crossing over, and chromosome mapping. So in the last video, we talked about the cross to determine if genes are unlinked or linked. And if they're linked, are they showing complete linkage or incomplete linkage? In this video, I want to explore incomplete linkage a little bit more and talk about crossing over and recombinants and recombinant frequency, how that can vary and what does it mean when it varies. And just explore in detail the main idea of incomplete linkage. So before we get into it, just a quick definition of recombination. And in this video, we're going to be focusing on recombination by crossing over for linked genes. But I wanted to remind you that recombination, formation of new combinations of alleles, can come about in two ways. One is for unlinked genes. Alleles can recombine into new combinations simply through the process of independent assortment. And then this is number 2 is what we're going to talk about in this video, recombination for linked genes by crossing over between non-sister chromatids during meiosis. So either of these will produce recombinant offspring, and that's what recombination is referring to. Okay, so this slide is somewhat reviewing incomplete linkage, and I want to give you a hypothetical example using numbers for a cross that might produce incomplete linkage. So we're going to do the typical diagnostic cross for linkage, dihybrid test cross, in which one individual is heterozygous for 2 genes, crossed to a tester individual. All right, what are the results from that cross? So in the Punnett square, we can set up this column with the gamete coming from the tester, and this column is having 4 different gametes coming from the double heterozygote. 2 of these gametes are going to be non-recombinant or parental types. Big A with big B, little a with little b. 2 of these gametes are going to be recombinant types, resulting from crossover in which now big A is linked to little b or little a is linked to big B. So these are those down here. All right, these are the recombinant gametes, and they're going to produce recombinant offspring. These are the non-recombinant gametes, and they're going to produce non-recombinant offspring. Okay, so that's indicated in this column, which are recombinant or non-recombinant. And by the way, you can use these terms interchangeably. The textbook, I think, uses parental and non-parental. I usually use non-recombinant and recombinant. All right, here would be some typical numbers you could see for a cross involving incomplete linkage. You would expect to see 2 parental phenotypes, either showing both dominant traits or both recessive traits. And they would be present in roughly equal numbers. In this case, for it's a hypothetical example, I made them equal. And if you sum these 2 non-recombinant classes, they would total to more than half, more than 50% of the total offspring. Here's the total offspring down here. Sum these two, 160 out of 200, that's going to be about 80, that's going to be exactly 80%. Okay? On the other hand, for the recombinant classes, you would also see 2. They would be either dominant for A, recessive for B, or recessive for A and dominant for B. They would be present in equal frequency, and if you sum them together, they would be less than 50%. All right, so the main conclusions: non-recombinant offspring, these here, are the 2 two phenotypes in roughly equal frequency and total more than 50%. Recombinant offspring, these here, two phenotypes in roughly equal frequency again, totaling less than 50%. All right, so using that hypothetical example, let's go through a calculation to determine what the recombinant frequency is from a dihybrid test cross. It's a pretty simple calculation shown here, and this is the same table from the previous slide, and here are the same numbers that were mentioned. So the recombinant frequency, RF, is the number of recombinants divided by the total number of progeny and multiply it by 100 to get a percentage value. So for this particular case, total number of recombinants, 40, total number of offspring, 20, times 100 gives you a recombinant frequency of 20%. The recombinant frequency is the percent of recombinant offspring resulting from this dihybrid test cross. Now, we'll be using both the term recombinant frequency, and that's expressed in percent, and the term map units. And the map units is simply the value of the recombinant frequency. So a recombinant frequency of 20% is equivalent to 20 map units. We'll be using this later, map units, when we talk about chromosome mapping. All right, so here's a feature of crossing over and the frequency of crossing over. For two linked genes displaying incomplete linkage, the frequency of crossover varies according to the distance these genes are on the chromosome. So crossover frequency correlates with the physical distance between a pair of genes. So let's look at three hypothetical examples. Two genes that are extremely close together. Let's say they're side by side, they're neighboring on a chromosome. What's going to happen for those two genes that are so close that they're pretty much next door? There's a very small chance for a crossover to occur between them. There's not much space for that crossover to occur. And so they would typically produce a recombinant frequency of 0. And that's what we see in complete linkage. In complete linkage, you don't see any recombinant offspring, only non-recombinants. Recombinant frequency of 0, therefore, is complete linkage. Let's look at a case where now genes are on the same chromosome, they're linked, but they're not exactly side by side. There's a little bit of space between them, and that's a little bit of an opportunity for crossover to occur. So genes that are linked but pretty close have a low to moderate recombinant frequency between them. And a third case would be genes that are linked, but now they're pretty far apart. And so the stretch of DNA between them is a large target for crossing over to occur. And so they would have a higher frequency of crossover between them. So let's take a look at what these would look like in terms of appearance on the chromosome. So here's the first example, two genes A and B that are linked, they're side by side, there's no space here, or very little anyway, for a crossover to occur. And that would produce, in a typical dihybrid test cross, a recombinant frequency of 0%. Complete linkage. Here is the second type, in which two genes C and D are linked, but there is some space between them. So this is a small, but enough of a target for some crossovers to occur, producing a low crossover recombinant frequency, in this case 15%. So this would be a case of incomplete linkage. And here's the third example now, two linked genes that are really far apart, and this is a big target for crossing over. So there would be a large, high frequency of crossover between these, and produce a recombinant frequency of 40%. All right, so that's just indicating the correlation between how close genes are together on a chromosome and the recombinant frequency that would result in a dihybrid test cross. All right, so now let's look at the situation, oops, sorry about that, situation in which there is recombinant frequency, so this would be incomplete linkage. And what's actually happening in all of the meiosis divisions in an individual that's heterozygous for two genes. All right? So an individual that's heterozygous for two genes, here's one homologous chromosome shown in red, another in blue. And this is illustrating what's happening in prophase 1 of meiosis. Let's say in this meiosis division, there is a crossover between A and B, such that crossover, remember, is exchange of segments between non-sister chromatids. The red chromatid here and the blue chromatid are non-sisters. All right, these two chromatids are going to be involved in the crossover, and they will generate recombinant chromosomes, big A linked with little b, or little a linked with big B. But notice also in this meiosis, there are two chromatids that are not involved in the crossover. And as these cells containing these chromosomes go through meiosis, they will generate two chromosomes and two gametes that will be non-recombinants. Two dominant alleles here, two recessive alleles here. So, the conclusion from this is, in a cell undergoing meiosis with crossover, only half of the chromosomes, and therefore half of the gametes, will be recombinant. Half of them will be non-recombinant. Okay? So what's happening in the meiosis divisions that don't have crossover? Here's a meiosis division in which there's not crossover between A and B. If we follow these chromosomes all the way through meiosis, it will generate 4 chromosomes and 4 gametes. All of these will be non-recombinants. So, let's consider the case of a hypothetical set of meiosis divisions in an individual that's heterozygous for 2 linked genes, A and B, that show incomplete linkage. And just for the sake of ease of calculation, let's say there are 100 meiosis divisions total. 10 of these are going to have crossover, and that's what's shown on the left here. And the rest, 90, will have no crossover, and that's what's shown on the right here. If there are 10 meiosis divisions, that's going to produce 40 gametes, right? Each division produces 4 gametes. And we just said that for a meiosis having a crossover, half of the gametes will be recombinant, these two in the middle. Half of them will be non-recombinant, this one on the top and this one on the bottom. So if there are 10 meioses producing 40 gametes, 20 of them are going to be recombinant, 20 of them are going to be non-recombinant. All right, the remaining 90 meioses will not have crossover. And what's going to result from that is none of these chromosomes and none of the gametes containing those chromosomes will be recombinants. So all of the gametes from these 90 meioses, 360 gametes total, will be non-recombinant. All right, let's take a look at kind of an accounting of these numbers on the next slide. All right, so this table is showing basically the same numbers that we saw in the previous slide and breaking it down into a table format. So of the 100 total meiosis divisions, 10 of them, 10%, have crossover between A and B. 90% do not. 10 have crossover, 90 don't. Of the 10 with crossover, that's going to produce 40 total gametes. Half of them are going to be recombinant, half of them are not. Of the 90 that don't have crossover between A and B, all of the gametes are going to be non-recombinant, 360. Okay, so if we sum up the number of recombinants and compare it to the total, that would be recombinant frequency. Total number of recombinant gametes, 20. Total number of gametes, 400. 20 out of 400 gives a recombinant frequency of 50%. Sorry, 5%. 5%. That's an error. Should be 5%. So the conclusion here is there's a difference in the percentage of meiosis having crossover, 10%, and the percentage of recombinant offspring, 5%. And this relationship is always going to hold. The number of percent of meioses having crossover is always going to be double the percent of recombinant that result from that meiosis in that individual. Okay? So here's an extension of that. Let's consider a case in which two genes are linked and they are extremely far apart on the same chromosome. So even though they're physically attached to each other by linkage, there's a huge target for crossover between them. And let's say the target is so huge that every single meiosis has a crossover. 100% of the meiosis divisions will have crossover, and 0% will not have crossover. So we can eliminate this because this is not happening. All of the crossovers have meiosis. If all the crossovers have meiosis 100%, then the relationship should be that half of the gametes and half of the offspring of a dihybrid cross will be recombinants. And so the recombinant frequency is 50%. 100% of the meiosis have crossover, producing a recombinant frequency of 50%. 50% is therefore the maximum recombinant frequency. And the reason for that is, well, you can't have more than 100% of the meiosis having crossover. So the maximum recombinant frequency, therefore, is 50%. So this brings up a point of consideration. And that point is the following. So we just went through the example shown here in number 1 for two linked genes that are very far apart on the chromosome, such that every single meiosis division produces a crossover that will give a recombinant frequency of 50%. But we would also see this same recombinant frequency for two unlinked genes. For two unlinked genes in a dihybrid test cross, half of the offspring are going to be recombinants by virtue simply of independent assortment. So, key point. If you do a dihybrid test cross, and that gives a recombinant frequency of 50%, you can't conclude if the genes are unlinked or if they're linked and very far apart. 50% is inconclusive for these two possibilities. Okay, I'm going to end this recording here, and next time we will talk about some specific examples of incomplete linkage, and just to work through some problems using recombinant frequency and predicting the results of a dihybrid test cross. All right, see you next time.